So our next speaker is Polina Mamoshina from DeepLongevity. And thank you very much for joining. Can you hear me?
Yes, I can hear you, Martin. Can you hear me?
We can hear you. Please go ahead and share your screen whenever you're ready.
Perfect. Yeah, thank you very much for inviting me. It's such a pleasure to be here alongside all the speakers. Just give me a second. I will pull up my slides. Yeah. So my name is Polina and I'm a chief scientist at DeepLongevity Limited. That's a spin-off company of Silica Medicine, which is focused on biomarkers of aging. And my today's talks will actually be about biomarkers of human aging. So a bit about the company. As I said, we're a spin-off of Silica Medicine. We were founded in Hong Kong in 2019. We focused on developing aging clocks and try to publish as much as possible. So right now we have a portfolio of those aging clocks and mortality predictors, and we also hold patents for some of them. We have a digital tool, which some of you may know and seen, that's Young AI. That's a platform that integrates our age predictors. And our main strength includes deep neural networks development and also genital adversarial networks. I also have to say that as a company, we're in a unique position that 70% of our staff is female, and we're also a woman-led business. So I'm really proud of it. So just a bit of a recap of challenges in human aging research. Of course, we can experiment, and a lot of talks today were presenting those results on animals, or going to present on animals. So we can do amazing experiments on animals, yet the translation rates from animal experimentation into human clinical are low. So depending on the therapeutic area, it can be as low as 86% failure rate for cancer, for example, for cancer therapies. So that's why we as a company more focused on tools that work on humans. And we are focused on developing tools that can measure biological age that is determined by physiology rather than chronology. So today, several of those are available, and this progress was started by Professor Steve Howard, who is going to actually present after me by his publication of hematological clocks in 2013. Multiple new clocks were proposed since then. That was mainly driven by availability of data. So those data types that we use to develop clocks are ranging from epigenetics to transcriptomics or even telomere length. At the same time, there is not only a revolution in data happening, but also a revolution in algorithms. So deep learning, it's a technology that's actually generated most of the hype for so-called AI. And in 2014, it started to change completely image recognition. Right now, in 2019, we believe it is actually changing healthcare. So why it's happening right now? So most of the algorithms were known since 1970s, but only now we are able to actually train them. So we have algorithms like backpropagation, others can actually allow us to train the deep neural networks. Plus we have computing power and we have big data. As Dr. Lee said, deep neural networks are exceptionally data hungry. So we actually need millions or thousands of samples to train them on. And what is also important, they're not only data hungry, but they are aimed to produce really accurate results. And regulatory agency, they already recognize the potential. So since 2017, FDA approved around 14 deep learning based tools for various applications. And for example, in 2017, they granted a fast track approval for Arteris cardio for the MRI heart extraction tool. And recently, quite recently, they approved atrial fibrillation detection developed by Apple. It's actually available right now in Apple iWatch devices in September. So since September. And for most of the cases, FDA actually granted fast track approval just based on the superior accuracy of algorithms. So regulatory is actually there. So they recognize the potential, and we believe that once we will be ready to enter clinical trials with the biomarkers of aging, if we will have biomarkers of aging that have been developed using deep learning, we will have an advantage there because, again, regulatory agencies, they are familiar with the technology and they should recognize the potential there as well. Since 2017, multiple deep learning-based clocks has been proposed and developed with our publication of deep hematological clocks. All of them range by accuracy, starting from really accurate facial image-based clocks that actually were developed by the company that just presented How AI, going to less accurate physical activity-based clocks. But why do we need so many markers? Why cannot we actually create a universal one? Well, apparently they measure different things. So this is an amazing study, and I kindly encourage you to check it out. It was published by Professor Dan Belsky and his group in 2018, where he compared biological age predicted by 11 biomarkers for people same chronological age. They were healthy, same chronological age. What he saw was really interesting. So while clocks are actually ticking, so they're working in the right direction, if we analyze people longitudinally, we see that they are getting older unless they are on interventions. At the same time, older chronologically and biologically. At the same time, if we try to analyze the ages predicted by those different aging clocks, we will see little or no correlation, even between methylation-based clocks. So even between clocks that are same data type. So that's why in a company, we believe in creating a really diverse portfolio of aging clocks so we can cover as many processes as possible. And that's why we not only working with really simple data types like, physical activity data extracted from wearable devices, but we also work with omics data a lot, including microbiome and transcriptome and so on. And the first clock I will present today is a deep hematological clock. That's a marker that is based on simple clinical blood tests. Well, first of all, I have to say that blood markers are not markers of aging by themselves. So if I analyze them with respect of age, if there are changes with respect to age, we will see that those common criminals like albumin tend to decrease with age, or cholesterol, glucose tend to increase with age. Yet it's not like a given number of, or level of albumin, we can actually predict how old the patient is. That's why we have to have a model that will capture dependencies in aging, in levels of those markers and aging. To do this, first collected a large data set of healthy people, and then we constructed a predictor of age, assuming that in healthy population, their chronological age should be close to their biological age. Of course, we will see variation, and our model is never perfect. That's why we have an error of 0.5 year, and also we have those outliers who are predicted older or younger than they chronologically are. And I will show you why it's really important to analyze those outliers in details with respect to any risks they may carry. Also, what is interesting with those models, while being rather accurate, when you try to analyze them on different populations, you will see that they actually fail to produce as accurate results as on the populations they were trained on. For example, if you have a model, if you have a deep biological clock that was constructed on Eastern Europeans and you try to predict age of South Koreans, they will be constantly predicted younger than they chronologically are, and other way around. So if you have a model that was built on South Koreans, they will be this model will predicting Eastern Europeans older than they are. At the same time, if we combine populations, we seem to solve this problem. So there's certainly a certain level of population specificity to changes in blood markers level, even for simple clinical parameters. If we analyze those people who are predicted younger or older, their chronological age by deep clocks, we will see that those who are predicted younger, they have lower hazard ratios or higher life expectancy. In other way around, those who are predicted older, they have higher hazard ratio for all-cause mortality compared to a normal group. So that means that we actually constructed a biological age clock. We also analyzed that whether their predicted age is more accurate than just chronological age by itself, because it's also good predictor mortality, and it's the case here. The predicted age by deep hematological clock is a better predictor. It also was a sex adjusted for us. We did the sex adjusted analysis in such case. At the same time, well, we also see that those lifestyle choices that know to accelerate aging and shorten lifespan, like tobacco smoking, for example, also accelerate age prediction. For example, smokers are predicted twice as old compared to their non-smoker peers at the same chronological age. But it's also interesting to see that clinical blood parameters themselves will allow us to predict smoking status using models. So there is, again, by analyzing the clinical blood tests, you can simply predict whether it's a smoker or not. Blood biochemistry, it's not the only data type we work with. As I said, we are trying to have a really diverse portfolio of aging clocks, and we recently published this paper with our as a part of our collaboration with Professor Vadim Gladyshev out of Harvard on deep microbiome clocks. What we showed was really interesting that first of all, using the whole genome sequencing, we can predict whole genome sequencing profiles. We can first generate the microflora abundance profiles, and then we can predict age of individuals, of healthy individuals with error rate of 5.9 years. What also interesting that we show that patients with type 1 diabetes are actually predicted older by their microbiome compared to, again, healthy controlled same chronological age. But what we believe those models are really powerful for is not only predicting biological age, but also experimenting with how the changes in input parameters will change the outcome or change the prediction of biological age. So this is an example we done for microbiome, when we analyze how the changes in each particular microbe here or bacteria here will actually change the biological age, whether it will increase the biological age predictions, means that this bacteria is so-called sena-positive, or decrease the biological age prediction, means that the bacteria is sena-negative. When we classify them in this way, we decided also to map them to their function in the body. And we saw that not all bacteria that are decreasing biological age are also having beneficial function. Some of them are actually pathogens, or those so-called youth infections, and other way around. So that's why we decided to introduce this, those 4 classes. And of course, the most beneficial one for us will be to promote those taxa that are decreasing age, but also have positive function in a human body. So to summarize, is that right now there is a big revolution in aging clocks that started in 2014. Multiple aging clocks has been proposed, but also there is a revolution of artificial intelligence in healthcare and in biomarkers of aging in particular. And the aging clocks, as illustrated, can not only be used to predict age, but also to identify those input parameters to models that are associated mostly with age. So it can help us future to modify input parameters and suggest what we actually have to change in patient profiles make them younger. So those are currently algorithms that we're working for, those sort of recommendation agents. So again, thank you for your attention, and I open for questions.
Thank you very much, Polina. Really interesting talk. I have some questions here in Slack. The top voted here is from Mario Vasquez Corte. Hey, Polina, how can AI models help towards completing testing, aiding the informational theory of aging? Feature extraction comes to my mind. What would be the input and target of the algorithms? Which are the easiest animal models to test in terms of data availability, easiness of use, and relevance? What about Sinclair's jellyfishes?
Right. Well, thank you. That's a good question. Actually, surprisingly, humans are the best model here, because if you analyze the data in the public domain, most of the data is available for human patients. Of course, you have restricted experimental capabilities there, but most of the data there is for humans. So it's not even for mice or C. elegans and others. So if we're speaking about especially the omics data. So probably humans will be the best choice here if we go there for deep learning, and we have to have a lot of samples there. As for the feature extraction, yeah, again, that's a powerful tool, but also I believe that when we have a model that can predict biological age, we also, again, can model how the input parameters will influence the prediction. So in this way, you kind of simulate the outcome without actually need to do the wet lab experiments. And that's provide us with a lot of possibilities here. We work with really different types of feature extraction, feature endpoint analysis models to understand how changes in features will change the biological age.
I agree with definitely that the humans are the best models here. I have also a question myself. So there's a large focus on getting the most accurate age predictor. But in reality, there is a large variation in how we age. So what do you think about the covariation in age prediction? So if you're predicted older, you're more likely to die, and if you're predicted younger, you're less likely to die. If we go for the most accurate clock, we may be missing that sort of clinically relevant data. What do you think about that?
Right, yeah, I agree with you. So the accuracy here, it's a bit misleading because, of course, when we analyze the accuracy of healthy population, we're just trying to be as close as possible to the chronological age, because we would expect the healthy population to have like a similar life expectancy here. And but it doesn't mean that it will work similarly on disease patient. So the accuracy here is not applicable. Plus, I mean, if you have you already have your chronological age in the past, so why do you need to have a predictor of it if it's so accurate? Plus, for example, animal studies on methylation analysis actually show that because those epigenetic-based clocks are really accurate predicting age, they also fail to show the results of biological age change upon interventions quickly. So, for example, there's an amazing paper, again, published by Vadim Valyshev quite recently, where they show that you have to put mice on a year of caloric restriction, which is 1/3 of their life, to actually see results in a change of their biological age based on their epigenetic profiles. So, of course, it's not acceptable in human practice. So that's why we not only need to be focused on like accuracy of prediction, but we also need to analyze how the clock will be able to reflect interventions, the association with mortality, also whether there are any conditions that are known to accelerate aging rate, or maybe lifestyle choices accelerate aging rate. So although other factors are also important when we evaluate the clock.
Very nice. Maybe one last question with a quick answer. So this is from Brian Chen. He's asking, with all different types of clocks, different models, different inputs, etc., what might it mean if the clocks don't agree?
It's an amazing question. We're trying to address it right now. I mean, to address it properly, we will have to have a really really rich data set with when we're on the same patient, we have preferably longitudinal different measurements for the same time points. So we really don't know now. So I have to say that we don't know. That is something we're trying to address. That's a really good question. Whether you being predicted older by your blood, which should probably reflect more like a systemic effect or acceleration of aging on a systemic level, be more, for example, were you predicting older by your fora and so on. So those are like really hardcore questions we're trying to address it now, but yeah, again, we don't have the data yet. So we will collect data and then we will see.
Very exciting. All right, I think we will move on to our last speaker. Thank you, Polina, for your presentation. It was really great.